we'll move forward with the next talk and come back for questions at the end. So Dr. Qinglan Ling will be presenting today on gene therapy development for SURF1 related Leigh syndrome. I think Dr. Hannaford, you have to stop sharing so that Dr. Ling can share their slides.
Am I sharing?
Yes, looks great.
Okay, good. Hi, everyone. Thank you so much for inviting me. And my name is Qinglan Ling, and I'm currently a postdoc at Dr. Stephen Gray's lab. And I'm sure a lot of you have heard Dr. Gray's talk about gene therapy that we do in our lab. All right. Why this is not. Okay, so first things first, my lab will start next year at UMass Medical School. So if anyone is interested in gene therapy for mitochondrial disease, please contact me. So this is my conflict of interest, and a part of the constructs or drug that we have designed and I will mention in this talk has been licensed to Teja Gene Therapies here at Dallas. And I'm sure you have all heard about previous talks about introductions of Leigh syndrome. So I'm not going to go too much details on this, but it is most common early onset mitochondrial disease, and it is progressive neurodegenerative disease with motor disorders and mental retardation. And there's some issues which Shamima have talked about a lot. And it can be caused by mutations in both nuclear DNA and mitochondrial DNA, which I've also heard. So, but I want to point out that pathological signatures of this disease can be very much different depending on what complex is compromised in the electron transport gene, the mitochondria. So SURF1 is the most common cause for Leigh syndrome, as we have heard from Alexandra. And it is ubiquitously expressed in the mitochondrial membrane protein encoded in the nuclear genome. The function of SURF1 is to facilitate HEA insertion. And it is critical for the cytochrome C oxidase complex or complex 4 assembly, which is right here. The fourth complex in the electron transport chain. And mutations of SURF1 lead to loss of function of the SURF1 protein, which then will lead to reduce the complex 4 or COX activity, and then will cause increased blood lactate as well as the SF lactate, and reduce the endurance capacity. So if someone tries to walk and run, you cannot last as long as someone has a functional complex 4. And I do want to point out here that according to the literatures that patients with SURF1 deficiency, they have only about 20% of complex 4 activity remaining. And this study is conducted with the muscle and fibroblast cells from the patient. So I don't think I need to give too much explanation about how the gene supplementary therapy work, and you have all heard about Dr. Stephen Gray's talk. But basically here, we are trying to introduce a functional copy of SURF1 delivered by AAV9 so that we can restore the function of complex 4. And to induce strong ubiquitous SURF1 expression, we have designed our construct for gene therapy. And the plan is to follow everything we've been done in our lab to use a single intrathecal lumbar injection. And our preclinical study has been testing on the SURF1 knockout mouse that Alexandra has also mentioned. And as he mentioned that this mouse do not show very, it's only show very mild behavior phenotype and even has a prolonged lifespan. So the only readout that we are using to evaluate the efficacy of our treatment is by looking at the restoration of the COX or complex 4 activity, as shown here in the publication in 2007 that generated this mouse model. The complex 4 activity is reduced to about 50% in different in different tissues. And if you remember that I mentioned earlier that the patient have about 20% complex 4 activity remaining, but here in the mice, they have about 50%. So we think this might explain why they only have very mild phenotype. So this is the design of our efficacy evaluations. And what we did is that we dosed the mice at when they are 4 weeks old and with different treatment plans. And basically we have a low dose and a high dose. And we also look at a combinational dose of intrathecal injection with intravenous injection to double the amount of vectors that we introduce into the mouse. And 1 month after the injection, which is when we will see the most protein expression, the time point where we see the most protein expression, we sacrificed the mice that collected tissues to look at the complex 4 activity, COX content level, and to evaluate if we have successfully expressed the SURF1 that we want to introduce. So this is the key data that got us excited about this study, that we, after the injection, we actually saw significant increase the complex 4 activity in the cerebrum and the cerebellum. Cerebellum is a little bit less than compared with cerebrum, but both of them showed partial restoration. And we also did a imaging study to look at the COX content level by using MTCO1 or COX-1 protein as the marker. And if you don't look at anything on this slide, just look at just look at this green circle outside. That's the level of the wild type animals. And then the red circle is the level of knockout animals with vehicle treatment. And all the animals that treated with gene therapy is in between the knockout and wild type, which suggests that we were able to partially restore the COX content level. And we then also try to correlate the COX activity as I showed in the previous 2 slides, and then the COX content as showed in the previous slide to see if they correlate with each other. And we did see a significant correlation. And this correlation, this relationship between COX content or COX-1 expression level and the COX activity has also been suggested in a previous study by Holman et al. In 2016. So this correlation further supported that our gene therapy was able to significantly restore the complex 4 function or activity. So then we wanted to see if we can get long-term efficacy from our gene therapy. So basically what we did is that we treated the animals with the same dose and everything. And then a month later, we put them on a treadmill. It's similar to a human treadmill, and then we'll let them run until the mouse are super exhausted. And we have a measurement to know that when they will be exhausted. So we also get the blood lactate before and after the running. And then the difference of those 2 data points are calculated as delta lactate. And then the same experiment were done when they were 10 months old. So this is more of a long-term evaluation. And after that, we sacrifice the mice and the tissue. And here, this is another data that got us very excited, that if you can see, especially at a 10-month-old, the red here is the blood delta lactate level for the knockout animals, and then the green is for the wild type animals. And our gene therapy was able to fully restore the lactate acidosis or elevated delta lactate back to the wild type level. So, so what I skipped here is that we then also looked at the complex 4 activity when they are 18 months old, and we were still able to see increase the complex 4 activity in the treated mice. But I want to quickly move on to what we are doing now, because the previous slides have already been published 2 years ago. And so basically what we did after that was that we evaluated the safety of the therapy, and it's safe in the mice. So then we moved on to a GLP toxicology study in the rat, collaborating with Charles River. And we dosed the rats with 3 different doses and using the same intrathecal lumbar injection. And then the tissues were collected at 7 days, 28 days, and 90 days after dosing. And then we conducted histopathology evaluations. So, and to our surprise, that when we look at the tissues, especially that at 28 days post-dosing, we saw some cytotoxicity or inflammation in some of the tissues in the rats, especially lumbar, especially dorsal root ganglia, and the lumbar is the most severe because it's the site of the injection. And I'm not going to go into the details of the slides, but I just want to show you that we saw cytotoxicity in the dorsal root ganglia, as have been mentioned in many other gene therapy studies, as well as in the cardiac muscles that we saw some inflammation signs, especially at the 28 days post-dosing. But this cytotoxicity is irrelevant to the doses that we give. So we think, well, it's since AAV9 has been tested in many other gene therapy studies, this might have caused by just we overexpress the SURF1, and that have given a lot of stress to the cells that have expressed too much of this protein. So then we decided to change what we did to change the design for our gene therapy. And this is the first version that I have presented a lot of data on. And then we adjusted the expression level of SURF1 that we got the three new versions, moderate, mild, and the lowest expression. And so the first thing we did is that we packaged all of this into AAV2 and we tested on patient fibroblasts. And here I really want to thank Casey for helping us to get the patient fibroblast. So here, this data, here we are showing that after infecting the patient fibroblast with our AAV2 vector, we were able to see a significant increase the complex 4 activity with all 4 constructs. So the green is the control cell line, and then the red is the patient line, and then the other 2 are the ones that infected with our AAV2. So as you can see here, with the reduced SURF1 expression level, we were able to see almost the same level of increased complex 4 activity. And this suggests that a lower transgene expression may exert a similar efficacy. And therefore, to reduce the possibility of toxicity, we decided to use this weakest. And then we fastly move forward to evaluate this new construct to see if it's effective in our mice. And again, I'm not going to talk too much about this, but if we can just look at this data here, that for as I showed before, that when we treat the mice at 28 days, we were able to see a significant increase of complex 4 activity in the cerebrum. However, here we were only able to see an increase when we treat them at post-natal day 1. And if you can imagine that the earlier you treat the animals, the more cells that you can transduce. And for cerebellum, we were able to see an increase in the animals that are treated at day 10. And the liver, it was the same at day 28. And the muscle, we were able to see an increased complex 4 activity with all treatments. So, and just to follow what we did before to look at the long-term efficacy. Here you can see that, again, at the 10 month, this is the knockout animals, this is the wild type animals, and our gene therapy, this new construct with the lower SURF1 expression, we were only able to partially restore the elevated delta lactate. So here we think that it is because that we reduced the level of SURF1 in some of the cells. So then this is showing a partial restoration. However, when we did another toxicology study in the rat, we did not see any toxicity. So we think this is promising, that is showing a partial restoration while it is safe. It will be safe. So to summarize, SURF1 gene therapy can partially restore COX activity deficiency in both CNS and peripheral tissues. And this effect can sustain for at least 17 months post-dosing. Again, I did not show the data for this, but I do want to point out that the effect can be long-term. SURF1 gene therapy can mitigate excess-induced lactate acidosis, overexpression of SURF1 that lead to cytotoxicity, and a better animal disease model would be critical to assess the potential correlation of restored COX activity and symptomatic benefits. Because here we are only looking at the COX activity, and you may ask, okay, would that help my kids to be able to walk, to be able to talk? And without a good animal model, we cannot answer that question. However, we in the last a bit over a year that we have generated a SURF1 knockout rat model, and we are currently evaluating this model, and so far the data look promising. So I just want to thank my PI, Dr. Stephen Gray, to give me this opportunity to work on this project, and it has definitely been a journey. And also my lab mate Matt and Yu Hui to help me with some of the work here. And my co-mentor, Dr. Prashant Misha, has taught me a lot about mitochondrial biology. And of course, Cure Mito Foundation, Casey has been with us since the starting of this project. Without Casey, and this project we will not have a presentation today. So if I can have any mission in my life, and treating this disease will be on top of it. And also some of the work that I have presented has been supported by Taysha Gene Therapies, and I want to thank the facilities and our collaborators.
Thank you,
And
Dr.
again,
Leigh.
my lab will be hiring next year at the UMass Trent Medical School. Please contact me if you're interested in gene therapy for mitochondrial diseases. Thank you.